Good morning, everyone, and thank you for joining us today for Garvan's first virtual public seminar. It's great to see so many familiar faces online, as well as some first-timers. Welcome to you all. For those of you who don't already know me, my name is Mara Jean Tilley, and I have the great pleasure and privilege of heading up the Garvan Research Foundation, the marketing and fundraising arm of the Garvan Institute. Today, we've got a fantastic panel joining us. We've got Professor Chris Goodnow, Professor Daniel Christ, and Dr. Ira Deveson. We're also joined by Priyanka, our wonderful public engagement superstar, who's going to keep us on track. We've got Joseph steering the meeting. If you do have any technical issues, please enter that into the chat and the team will do their best to help you. So before we kick off, I'd like to begin by paying my respects to the Gadigal people of the Eora Nation, the traditional owners and the custodians of the land on which the Garvan Institute is located. We recognise their continuing connection to land, waters and culture, and pay our respects to elders past, present and emerging. I'd also like to acknowledge and sincerely thank the many thousands of healthcare workers working in our hospitals and clinics, many individuals on the front line there, including some of our Garvan clinician scientists, and of course our dear friends and founders at St Vincent's Hospital. Our thoughts are with you all. So it is great to see such a fantastic turn up to this morning's public seminar. And I think that certainly for all of us at Garvan, we are reassured that despite the unprecedented crisis that we currently face with COVID-19, what we do recognise is that medical research has never been more important, and the support of our Garvan community, all of you with us today, has never been more critical, and we sincerely thank you for your enduring support of Garvan's research. In terms of the format for this morning, we've got 3 key speakers, followed by a live Q&A. So for those of you unfamiliar with the Zoom function, if you look down in the centre of your screen, you'll see there's a Q&A button where you can enter in live questions. You can do this either anonymously or with your name, but I'll probably refrain from mentioning names just to protect everyone's privacy. So, without further ado, I'm delighted to introduce the Executive Director of the Garvan Institute of Medical Research, Professor Chris Goodnow. Chris is an internationally renowned immunologist, and fortunately for us, we've got Chris really leading Garvan's rapid research response to COVID-19, and so he's going to introduce that work to you before we move over to our other panelists to talk about their specific research projects. So please join me in welcoming Professor Chris Goodnow. Chris, over to you.
Sorry, I was muting myself. Welcome everybody. Thank you, Mara Jean. Welcome everyone to the Institute virtually. We wish you could all be here physically. We will get there again, but there are some advantages, I think, to technology, if nothing other than it allows everyone equal access to information and to keep your finger on the pulse of what's happening at Garvan. Let me tell you, we're going to hear a little bit from 2 of our real star researchers, Professor Daniel Christ and Dr. Ira Deveson, in a little while. So I'll just take a couple of minutes to set the scene if I can, and just remind you a little bit about what we know and what we don't know about COVID-19 and the virus that causes it and the immune response against it. So as you know, COVID-19 is a disease. It's caused by a virus, a coronavirus, that has leaped from bats through an intermediate host that hasn't yet been identified into the human population. It's not the first time that's happened. It happened in 2003 in the SARS epidemic, a very close cousin of this virus, in that case through civet cats. And it's happened many other times. It's happened through camels, from bats to camels to humans in Saudi Arabia with another disease similar called MERS. And in fact, most of the great epidemics and pandemics throughout human history have been from viruses leaping, or bacteria leaping from animals into humans. Now, the key problem that we have with this particular virus is its incredibly variable effects in each of us. So some of us, as you know, develop no symptoms. We don't even know we're shedding the virus, what we call asymptomatic transmitters. Others, a mild disease, not that different from a common cold or a flu. But then there are some, a good fraction, up to 15%, who develop very severe disease that needs care in hospital, and around 5% who need critical care in an intensive care unit. And of course, the absolute terrible end of the spectrum is all of the deaths that we've seen around the world. Now, this is something that happens every time a virus jumps into a new species. It's not well attuned to our immune system, and our immune system tends to over-respond. And that is probably at the heart of why some people get very severe disease, that essentially it's not so much the virus itself that's making us sick, it's actually our cells, our immune reaction to the virus that's destroying the lungs, that's creating clotting in the vessels, that's damaging the heart and many other organs, and that's an issue. The other big issue is the variability because of this variability in the way our immune system responds. There's a variability in the extent to which we make antibodies, and that's going to be a problem both for natural immunity. Some of us may develop it for a while, others won't, and it'll be a problem for vaccines. And so for that reason, one of the big projects that we activated very early on, and Daniel Christ is leading this, and we'll tell you about this, builds on 20-something years of experience of Daniel developing and genetically engineering monoclonal antibodies to essentially achieve what you'd like our own immune system to achieve, these perfect homing missiles that latch onto the virus and never let go, but bypass all that complexity of variable immune response and get straight to what we really want to have in our bloodstream to protect us from infection or to clear the infection once it's got in. Now that probably is enough of an introduction from me. As you know, Garvan has enormous expertise in genomics and in the immune system, the immune response to cancer and inflammatory diseases, healthy aging, and every part of the Institute in one way or another has come together to pitch in to help crack this problem. So what you're hearing about today are just two of the most advanced projects, but there are many other ways that the Institute is playing a role while still continuing to chase and advance all the research in all those other diseases that we care so much about. So I might hand over to Daniel Christ at this stage via Mara Jean. Thank you.
Thank you very much, Chris, for that introduction. Before we hand over to Daniel, I just wanted to ask a couple of questions, if I may. Why is Garvan mounting such a significant effort against COVID-19, given that we've not previously undertaken research in infectious diseases?
Yeah, thanks, Mara Jean. That is a really good question. As you know, the Institute has a unique portfolio. One of the things that makes us very special is the way that we bring together research in cancer, research in immunity and inflammation, research in diseases of aging, for example, of the bone or diabetes, neurodegeneration. But the thing that really ties it all together and breaks down all of the silos and barriers is genomic technologies, DNA technologies, massively parallel DNA sequencing. Now, you know, some people probably don't think of the Garvan as a research institute that works on infectious diseases, but in reality, we've been working on them for a long time. So just to take one example, Professor Stu Tangi, who leads an Australia and New Zealand-wide consortium of clinical immunologists and geneticists and basic immunologists, he he's done a lot of beautiful work, and that team's done a lot of beautiful work explaining why some children develop devastating immune reactions against viruses and bacteria that for most of us really are mild or asymptomatic. And so they've been able, through genomic technologies, to pinpoint why the immune system is under or over responding to the virus, and in some really spectacular cases, to actually use that information to replace what is missing in the immune system. If the immune system's over responding, to actually give back the missing break on the immune system as a targeted biological, a monoclonal antibody drug, for example, to completely cure the disease. So we've been in this area for a while, and that's why we really felt, while we don't want mission creep, we got a lot to contribute to tackling this big problem.
That's fantastic, Chris. Thank you so much. So everyone, our next speaker is Professor Daniel Christ. Daniel is head of the Antibody Therapeutics Laboratory and director of Garvan's Centre for Targeted Therapy. He and his team are engineering antibodies for COVID-19 protection and therapy by targeting the surface proteins of the SARS-CoV-2 virus, the novel coronavirus that causes COVID-19, which the virus needs to infect human cells. Daniel, welcome. Over to you.
Thank you, Marilyn, and good morning to the audience. Let me just start this. All right, so antibodies are produced by the immune system to fight bacteria and viruses. But over the last 20 years, they've also become an important class of human drugs with over $80 billion sales in 2019. And those sales are mainly in cancer and inflammation, but also in infectious disease. And their market share is sort of increasing rapidly. And they now constitute, in fact, a large and sort of growing proportion of the overall proportion of drugs entering clinical trials and also increasingly clinical practice. So in fact, 6 out of the top 10, 12 selling drugs in 2019 were monoclonal antibodies. And sort of examples include the anti-HER2 monoclonal antibody Herceptin for the treatment of HER2-positive breast cancer, as well as Humira, which is in fact the world's best-selling drug with around $20 billion sales in 2019. And this is an anti-TNF-alpha drug, which is predominantly used for the treatment of moderate to severe RA. And in fact, this drug was developed out of my former lab at Cambridge University. So importantly, antibodies are also powerful weapons in our fight against infectious disease. And just to mention Synagis, which is an antibody against respiratory syncytial virus, or RSV, which is a leading cause of pneumonia in infants. And this antibody is given to at-risk infants by monthly injection during the RSV season. And that includes both preterm babies, so effectively everyone born before 29 weeks of gestation, and also those with immunodeficiencies. So how do we actually make these drugs? So the development of antibody therapeutics is actually over 100 years old, and it goes back to Emil von Behring's work on the development of the diphtheria antitoxin.
Our public
And it was done by immunizing horses
not live streaming
with the toxin, and then sort of a couple of weeks later, the horses were bled, serum was isolated and injected into patients. And this was in fact very effective. I mean, the mortality rate in infants for diphtheria dropped from around 50% to more like 1 to 5%. But the treatment also could have considerable side effects because of the animal origins of the antibodies. So these days, we rarely use animal serum and animal antibodies, although there are some sort of exceptions. I mean, for bespoke applications, for instance, the snake antitoxin antivenoms developed by CSL and others. Instead, we produce fully human monoclonal antibodies in the laboratory. And these antibodies have a single or what we call monoclonal specificity. And this sort of enables the generation of sort of defined medicines without batch-to-batch variation. So mouse monoclonal antibodies were developed in the 1970s by Cesar Milstein and George Köhler. And then the technology was later expanded to fully human antibodies by my former PhD advisor, Sir Gregory Winter. And these advances were awarded the Nobel Prizes in Medicine in 1984 and Chemistry in 2018. So why have antibodies become such an outstanding success story? And it really goes back to their properties. So antibodies have very favorable properties compared to traditional chemical drugs, or what we call small organic molecule drugs, such as aspirin. In particular, they have very high binding strengths, or what we call affinity. And in fact, the affinity is often about 1,000-fold higher than that of traditional chemical drugs. And that also has the direct effect that these drugs tend to be extremely specific. So they will bind to your target, but nothing else. They can have a very long serum half-life, so in the range of weeks to months. And in fact, some of the recent antibody drugs currently under clinical developments can have half-life up to about 6 months, which would enable, for instance, to inject the drug only once during a flu season or during a pandemic outbreak. And it's really the combination of these factors that results in an overall very high success rate. So the success rate of monoclonal antibodies in development is almost twice as high as that of traditional chemical drugs. So a range of antibodies have been described against coronaviruses. And in particular, several highly potent and also extremely well-characterized antibodies were described after the 2003 SARS outbreaks. And these antibodies bind to a part of the virus called S protein, which is critical for sort of binding to cellular receptors on cells, which facilitates the entry into human cells. So these anti-SARS antibodies have been shown to effectively block the infective process in vitro, and also importantly, to provide sort of protective immunity to animals when challenged with an otherwise sort of lethal dose of the SARS coronavirus. And sort of one important thing to notice is that the binding sites of the antibodies on the coronavirus spike proteins are extremely conserved and similar between SARS-CoV, so the causative agent of SARS, and SARS-CoV-2, the pathogen causing COVID-19. And this sort of similarity, which to us suggested, and it's sort of in the range of 75 to 90%, which to us suggested that many of these antibodies might be cross-reactive between SARS and COVID-19, sort of inspired us to sort of further develop and engineer these well-characterized antibodies, not just against SARS, but also against COVID-19. So we've developed a pipeline at Garvan, in which we sort of feed antibody sequences from the 2003 SARS outbreak, but also antibodies cultured from laboratory libraries, which we had existing in the laboratory, as well also increasingly sequences from antibodies from recovered COVID-19 patients. And this antibody pipeline already contains over 100 million different antibody variants, which we are now screening for increased affinity or binding strengths. And then the top candidates from these screens are tested for increased neutralization of the live virus. And this is then followed by clinical studies to determine safety and efficacy. And this work is carried out in collaboration with our colleagues at the Kirby Institute under sort of biosafety PC3 conditions. So the overarching idea here is really not to start from scratch, but really take all the existing information we have. So antibodies from SARS, antibodies from existing libraries within Garvan, antibodies from recovered COVID-19 patients, and to sort of repurpose and re-engineer. And the analogous example here is really something like Apollo 11, where sort of critical oxygen system failed halfway through the mission. And the crew really only had a couple of hours to sort of assemble something out of existing parts and duct tape. And it probably wasn't perfect, but it was certainly sufficient to get them back to Earth. And really importantly, it was fast and it was a relatively low risk approach. So this approach is already generating a lot of very promising data. And I mean, just to show you something that's really just sort of fresh out of the lab this week, here you can see sort of stepwise enrichment of antibodies over 4 rounds of selection, where we sort of simultaneously drop the target antigen concentration of the COVID-19 spike protein into the picomolar range. And here you can see sort of stepwise enrichment of antibodies against the SARS-CoV-2 spike protein, as shown here in green, but not control antigens. And we can see a signal down to the picomolar range, which is exactly what we would like to see in a potential antibody therapeutic. And this is incredibly dilute. And just going back to my previous comments about these drugs potentially being up to about 1,000-fold more potent than traditional small molecule chemical drugs. And this work was carried out by Orhan Matsigi in my lab, who's a very talented PhD student. Okay, just to summarize this presentation, so antibodies are an extremely well-validated class of human therapeutics. They can be produced in the laboratory in vitro, completely circumventing the immunization of animals. And they can provide passive immunity for both therapy and protection. They are a relatively safe and low-risk approach, and this is exemplified by the development of Synagis for RSV, for which, in fact, we still do not have a vaccine, even after decades of development. Antibodies can be given to at-risk individuals. I mean, this includes the elderly, chronically ill patient, and also our healthcare workers on the front line. And sort of using all the available current information, we have developed sort of a pipeline of over 100 million different antibody variants, which we're now screening against SARS-CoV-2 in order to identify antibodies with increased target affinity and then also, importantly, increased neutralization potential against live virus. Okay, I'd like to thank my people in my team at the Garvan Center for Targeted Therapy. I mean, who've really been working countless night shifts on this. We've worked on this for less than 3 months, and we've really made amazing progress on this. I'd also like to thank our generous donors who've really made this work possible. And your continuing support is really going to be absolutely essential for the overarching goal of this research, which is really to have these medicines ready for clinical evaluation later in the year, as in the 4th quarter of 2020. And I'll thank you for your attention.
Thank you, Daniel. That's really fantastic. I'm just going to jump in and ask you a couple of quick questions, if I may. Now, forgive me, but can we take a step back and can you tell us exactly how antibodies work?
Yeah, so antibodies are part of what we call the humoral part of the immune system. So the immune system has sort of 2 branches. It's cellular based, which is sort of T cells and NK cells, and it's a secreted part, which involves antibodies. And they are secreted by a type of cells, which we call B cells, right, which is sort of stimulated by antigen. And then the cells go through a maturation and switching process where they then can churn out, they turn into sort of antibody factories, and they can churn out really like grams and grams of antibodies over many months to week. And these molecules have very fascinating properties. I already mentioned their extremely high binding strengths, right? About 1,000-fold higher than traditional drugs. They have very long serum half-life, and they also have very powerful immune functions, as in that they can, for instance, clear viral debris, they can activate other parts of the immune system, and it's really this sort of package. Very high affinity, very high specificity, incredibly long serum half-life, which we can further increase through engineering approach. technologies, which is going to be incredibly important for like a pandemic like this, because we're going to be limited in production capacity. We're going to be limited how often people can actually go to their GP to get the injection. We might be able to give this only once a month, once every 3 months, possibly once every 6 months or so. That really comes down to these quite unique package of properties that these antibodies do possess.
Thanks, Daniel. And just to clarify, we would typically make antibodies naturally in reaction to a potential virus. But what you're doing, if I understand correctly, is engineering the antibodies at scale. Is that correct?
Correct? So we use a technique called phage display, which I mentioned was the basis of the 2018 chemistry Nobel Prize for my advisor, Gregory Winter, and this is a complete in vitro technology. So we can make hundreds of millions of variants in the laboratory, and then we can use sort of a type of laboratory evolution to pick out sort of the winners of this sort of selection process. So those with the highest binding strengths, the highest target affinity. And that process has the huge advantage that it's incredibly fast. So we can do iterations in like 2 to 3 weeks. It's completely in vitro, so we have control over the conditions. We can exclude certain properties which we consider undesirable. We can include others which we want, like for instance, broad specificity against coronavirus. It really gives us the opportunity to fast track the process and enables unprecedented control over conditions which you just do not have when you immunize animals, or also when you're developing a vaccine in the general population, which might have very varying immune responses.
Great. And so you mentioned your collaborators at the Kirby. When are you hoping that the clinical trial will start at the Kirby?
So we would hope to start the clinical trials in the fourth quarter of this year.
Okay, exciting. Thank you, Daniel. Fingers crossed. Okay, guys, so we're going to move to our next speaker now, Dr. Ira Deveson. So Ira is the head of the Genomic Technologies Laboratory at Garvan within the Kinghorn Centre for Clinical Genomics. He and his team are tracing the evolution of COVID-19 by sequencing the virus's genome in infected patients to detect genetic variations that may provide critical data to inform Australia's response in real time. Ira, welcome.
Thank you. Thanks for the introduction. I'll just load up my slides. Cheers. So as mentioned, I have the pleasure of leading Garvan's Genomic Technologies Lab. And in the Genomic Technologies Lab, our goal is basically to develop and to test and to optimize new technologies for genomics. So we have a lot of experience in DNA and RNA sequencing, which of course is the very sort of core of genome science. So we're routinely working on sort of developing new molecular and computational tools to basically improve our capabilities in this area. And I guess until a couple of weeks ago, our work really had nothing to do with viruses. If I'm being perfectly honest, I still don't really know all that much about viruses really, but this coronavirus pandemic that we're living through is a pretty extraordinary event. There's obviously millions of lives at risk around the world. And so as a scientist, and I guess as head of the Genomic Technologies Lab, I had to sort of ask myself what we could possibly do to help. How can we utilize our resources and our expertise in genome analysis to potentially contribute in some way to Australia's response to the coronavirus pandemic. So when we sat down and thought about it, we realized that there were a bunch of things we could do, some of which could potentially turn out to be quite helpful. Of course, we're in the midst of a very fast-moving and extremely complex challenge, but I guess the best thing we can do is to sort of try to think creatively and come up with some innovative solutions to the problems that we're facing. So we're trying out a whole bunch of different things, but I'm just going to quickly tell you about two of these projects where the work is well and truly underway. So by way of background, and Chris touched on this, is there's a particularly problematic thing about coronavirus is this observation that basically up to 50% of individuals infected with coronavirus do not show any obvious symptoms. It may be even higher than that. Now that I guess sounds like a good thing in principle, but the problem is that these people go about their daily lives, and they can spread the virus around, pass it on to other people without ever actually knowing that they were infected. What kind of a lot of early studies have indicated from China and elsewhere is that the majority of patients with COVID-19 that we do detect were in fact infected by an individual whose disease was never detected. And I mean, there's some modeling suggests that maybe only 5% of infections worldwide have actually been detected to date. Now, these numbers are pretty crude. They're sort of based on modeling, and they're not certain. But the take-home message is very clear, and that is that to get on top of coronavirus, we need to do more testing. We need to test more individuals, and critically, we need to start testing asymptomatic individuals. Or, I guess, to put it simply, we would really like to just be able to test everyone all the time. That's how we would really get on top of this. So what's stopping us from doing that? Like, you know, we know how to test for coronavirus, so why can't we just do lots of it? Why can't we just do more testing? I guess one problem is that the current test we have, you essentially take a sample from one patient. This is typically a nasal swab, and you put it in a test tube, and then you do some sort of biochemistry on that in that test tube, and it gives you an answer, positive or negative. You know, does this patient have the virus? This is you know, all very well, having this one patient, one test. It's all very well if you are sort of testing tens or maybe hundreds of patients. But if you're trying to test thousands of patients, tens of thousands, maybe hundreds of thousands of patients per day, this one test, one patient system means that you're going to just require a huge amount of labor, a huge amount of materials, huge amount of enzymes. It's going to be expensive. It's going to be cumbersome. So this one patient, one test system just really doesn't stack up. It's not feasible for those kind of numbers. So what we're really interested in, and what we would like instead, is a test to be able to test multiple patients, ideally thousands of patients, in a single test tube. So a single reaction, and get an answer simultaneously for everyone. So this is something that we've decided to try and work on, basically developing a massively pooled or massively parallel diagnostic test for coronavirus. So to understand our idea, I'm just going to quickly explain how the current test works. What you do is you basically take a sample from a patient, you perform a molecular process called PCR, and in this process, a specific little piece or a little section of the coronavirus genome, which I've highlighted in blue, gets duplicated rapidly. It gets rapidly amplified, building up more and more copies of that DNA sequence in your test tube until it sort of passes some threshold for detection. And then we can say, yes, this patient does have the virus. If there was no virus present, that DNA sequence doesn't get amplified, and so we don't detect the virus. So this is cheap, it's fast, it's very reliable, but as I said, it's effectively limited to one patient for one test. So we don't want to reinvent the wheel. We just want to sort of develop a way to basically parallelize this process. So what we've done to do that is develop a set of special DNA barcodes. And what we do is incorporate these DNA barcodes into the coronavirus DNA sequence that we're just about to amplify. The barcode is basically unique to, a different barcode is unique to each individual patient. And what that means is that we can take material from lots of different patients and pool them all together into a single tube. And then we can just perform one single amplification reaction on that pooled sample. We then take advantage of modern DNA sequencing technologies, which allow us to read all of the DNA molecules in our tube at once. And when we do this, we obviously detect a whole lot of virus DNA. But the crucial thing is that we can read those sample barcodes that were incorporated at the start of the process. The number of times we read or count each barcode is basically proportional to the amount of virus that was in that patient sample at the start. And crucially, if a patient didn't have the virus, then no barcode got incorporated. And so we don't read a barcode for that patient when we do this DNA sequencing. So we've just completed a pretty promising little pilot experiment in which we used 192 of these DNA sample barcodes to basically test for the presence of virus in 192 samples, which were in fact samples where there was a known infection. And the results are pretty promising. They sort of demonstrate that our test is pretty accurately able to measure the abundance of the virus in those samples. But the beauty of this system is that we can quite easily make many more of these sample barcodes. We can make as many as we like, and it's quite realistic that we could potentially be able to test 1,000 or more samples in one single reaction or one single tube. And I guess the big picture here is that in order to sort of relax the lockdown and allow people to kind of go back to work and go back to their daily lives, we need to be able to routinely test large sections of the population so that we can quickly pick up and quarantine any potential new outbreaks. And if we're going to be able to do that, we really need some highly scalable methods like this one that we're attempting to develop here. So that's just a simple diagnostic test. And just like the current diagnostic test, which I've shown you, we're just measuring a very small section of the virus genome. We're just a small chunk of a single gene. And so that tells us whether the patient has the virus or doesn't have the virus, but it really doesn't tell us much more than that. So we can actually do quite a lot better than this. In my lab here at Garvan, we're equipped with a set of really cutting edge DNA sequencing devices called nanopore sequencing devices. A couple of these are pictured here. And using these, it's actually relatively straightforward for us to read the complete genome sequence of coronavirus that we've isolated from an infected patient. So the virus is 30,000 bases long, which are 30,000 DNA letters long. It encodes 11 different genes, and we can basically read and reconstruct this entire sequence in really just a few hours, which is obviously pretty cool. It's pretty impressive. I guess the question is, why would we actually want to do that? The answer is that as the virus replicates and spreads from host to host, it accumulates minor changes or sort of typos in its DNA sequence, which we call mutations. And these accumulate throughout its genome sequence. So if you compare the sequence of a virus from one patient to another patient, you'll actually find that they're slightly distinct. They differ by a handful of letters or a handful of positions in the genome. Crucially, also, when a virus is passed on from one patient to another, the changes that virus inherited are similarly passed on to the next patient. And so what this means is that if we sequence the genome in a whole lot of different patients, we can use the set of mutations that we identify to basically construct like a family tree for coronavirus. So at the root of this tree, we believe, is this single case in Wuhan, China, a single infected individual from whom all subsequent infections are ultimately descended. And as the virus has sort of spread out across the globe, it's basically diverged into different branches or lineages or families in different parts of the world. And so when we sequence any new infection, we can actually place that new infection somewhere within our family tree. So the genome analysis allows us to determine basically, how closely related is the virus in patient X to the virus in patient Y? Is it the sister virus, the cousin virus, the second cousin virus, so on and so on. So we can use this information to sort of track the virus, so infer the origin of new infections and observe how it is spreading through our community. So by tracing transmission events, we can potentially answer questions like, how many people does the average infected patient pass the virus? And we can potentially identify super spreaders or people with abnormally high transmission rates. We can potentially even be able to ascertain the sort of most frequent modes by which community transmission is actually taking place. So questions like, do most people get infected at, in their workplaces, on public transport, at family barbecues? You can actually sort of dig down into this information, which is going to be really powerful for tracking and controlling the coronavirus pandemic. In addition to this information on viral surveillance, the genome sequencing is also going to allow us to study viral evolution. So we'll sort of be able to see how quickly the virus is mutating and changing. We can identify potential new branches or potential new families, I guess, that are more or less infectious than others. And likewise, as new drugs and vaccines come on board, we'll be able to determine whether different sections of this family tree, different lineages, are equally susceptible or potentially resistant to those drugs and vaccines. And so this is going to be really powerful information as the pandemic progresses. So our lab at Garvan is now receiving and collecting samples from various hospitals in Sydney, and we're generating complete genome sequences for coronavirus in different infected patients. We've already basically sequenced almost 100 full viral genomes, and we're really just getting started on this. This is not something that we're doing on our own, and a really key point here is that it wouldn't actually be all that interesting or useful if it were just us, because the data is only powerful if we're able to sort of compare this with similar data that's being collected by labs around the world. And so it's really fantastic then that there's already a large network of labs all over the world who are sequencing the coronavirus genome, sharing these sequences, and contributing to this effort to create a massive family tree for coronavirus. So here I'm just showing you what that actual tree looks like, with more than 3,000 genomes sequenced today, although this is actually now out of date because every day more and more genome sequences are being added to this tree. And I think it's quite an inspiring thing, I guess, that many people working on this and contributing to this network, had basically no interest in viruses until a couple of weeks ago, but have been able to sort of rapidly redirect their, I guess, resources and interests into this collective mission. So this is very much the case for my lab, where these two goals that I've shown you, I guess, this idea of developing a test that can simultaneously detect coronavirus in potentially thousands of patients, and the idea of tracing the transmission and evolution of coronavirus with modern genome sequencing. These goals have become very much central to my lab, the mission of my lab, and the work that we're doing every day. So I'll leave it there and finish up by acknowledging the hard work of the people in my lab who are working on this: Jill, Igor, James, and Hasindu, and also two other researchers within Garvan, Manu Singh and Jim Blackburn, who are collaborating with us on this. Also, our key collaborators at the Kirby Institute and within New South Wales Health Pathology, who are collecting and processing these samples that we're analysing in our lab. So the work obviously couldn't happen without them. And also, the work is obviously supported by generous donations from philanthropy and also from the grant bodies within Australia. And so I need to acknowledge them. It wouldn't be possible without that. So also, thank you everyone for tuning in to listen. I hope you've enjoyed the talk. Cheers.
Thank you, Ira. That's really cool work. Thank you very much for sharing it. In terms of the genetic variability of COVID-19, at the moment, are you seeing indicators that it is more likely to mutate than other viruses, or less likely, or is it too soon to say?
So the virus is mutating already. It with every new transmission between patient to patient, it accumulates one or two little changes in its DNA sequence. But actually, the mutation rate of coronavirus is quite low compared to other viruses. So it's much lower than, for instance, influenza, which mutates very rapidly. And this is, I guess, a really good sign for us for the potential durability of a new drug or a new vaccine. It means that while it may not last forever, it may last more than one season. It may last for a long time. And that's in contrast to, for instance, flu. Because of the very high mutation rate in flu, we need a new vaccine every season. So the fact that coronavirus has a low mutation rate in that sense is a very good thing.
Fantastic. And just thinking about the team at Garvan, are they safe looking and working on the genome of COVID-19?
Absolutely. So my lab actually doesn't handle any of the patient samples directly. Those samples are collected at various hospitals, and then they're being sort of processed at the Kirby Institute in their specialized facilities for handling these things. That upstream process involves extracting RNA from the virus sample. Once that RNA, which RNA is basically a molecule just like DNA, once that's been extracted from the virus, it's completely inert. It's no longer infectious, and so it's perfectly safe for us to handle, which is very, very helpful for us.
Yes, that's very reassuring. Thank you, Ira. All right, I think we might bring the panel back online. So guys, if you can activate your videos, please. And I'm going to turn to the live questions that we've received via the Q&A function. There's some really good questions here. I'm going to try and get through them as quickly as we possibly can. So Chris, the first one's for you. Has the Garvan received special COVID-19 funding for the COVID-19 research that Garvan is undertaking?
Thanks, Mara-Jean. We have had funding from University of New South Wales, which is internal competitive funding. We obviously are redirecting effort wherever we can, where there's something to contribute, as Ira's described, or in Daniel Christ's lab, which is supported by a large NHMRC program grant for therapeutic antibodies in part. And we've got grant applications in with the Medical Research Futures Fund that we are optimistic of. But, for both these projects, particularly, so far we've been able to, put together existing resources to get these things to such promising points. But to scale up to the levels of, so in Daniel's case with the antibodies, the big ticket item will be the scale up production of the antibody as quickly as possible. And that is not yet funded, but we're obviously working on that. It'll be crucial to have that funding and have that not be a thing that holds up progress.
Thanks for that, Chris. And I can also confirm that we're working with our friends and partners at the Kirby to launch fundraising appeals for both projects too. So that will be out in market very, very soon. So thank you for that. The next question's fantastic. If the immune response to the virus can be the reason some people suffer greatly, what is the situation for people such as those who have been treated for cancer, whose immune system has been compromised? Chris, do you want to tackle that one?
Sure, and then Daniel might want to jump in there. The short answer is that from all the work that's been published around the world so far, that it's not a good thing if you have a compromised immune system because of cancer, or diabetes, or lung disease. All these things seem to increase the probability that you're on the more severe end of the spectrum. Exactly how that works isn't known, because at the same time, for people with severe COVID-19, a lot of them are being given broad spectrum immune suppressive drugs, for example, steroids. And it's not clear that that's helping or hurting. And so this is the double-edged sword problem that physicians have and that we as scientists have. It's the same problem we have for kids, the kinds of kids that Stuart Tanguy and his Circa Consortium study, that on the one hand, one part of the immune system is over-responding and you want to suppress that. But at the same time, another part of the immune system may be under-responding, and that might be part of the reason why the virus isn't being eradicated. So what really needs to happen is we've got to cut through all of the distractions of the many things that are going on in someone with severe disease, get to the root cause of why is the immune system over-responding, and fix that with a very targeted treatment, probably a monoclonal antibody.
Thanks, Chris. Daniel, did you want to add anything to that?
Maybe just to mention, supports Chris' view here. There's very little data, and I think the bottom line at the moment that if you have cancer or an immunodeficiency, you need to be very careful and practice social distancing, and that's also a personal message. My father, for instance, has prostate cancer at the moment and is undergoing radiation therapy. You need to be very careful.
Is he watching us this morning, Daniel?
I think he's in Europe, so I think it's like probably 3 o'clock in the morning or so. I doubt it.
We'll send him a video afterwards. The next question is, I think probably for you, Daniel, do antibodies require the same degree of trial verification as vaccines?
So one of the unique properties of antibodies is that they're actually about 98% identical to each other. So they only differ in what we call complementary determining regions. So it's the bit that interacts with the actual target antigen. And that has sort of the effect that they actually have fairly similar properties to each other, right? And in contrast, for instance, we've never made any successful vaccine against
any sort of coronavirus, right? So just to answer your question, so yes, although these antibodies will have to go through careful clinical evaluation, this is a highly streamlined process for which we have lots of experience, and that's just simply not the case for our vaccine development efforts against coronavirus.
Okay, thank you for that. Would a COVID-19 monoclonal antibody work in a person with B-cell antibody deficiency? Another
Absolutely.
one for you, Daniel.
And it's one of the great advantages of antibody therapy. It's passive immunotherapy. It's really quite independent of the age, the status of the patient, and it can also potentially be given both preventative and therapy. It's a unique advantage of this passive approach. You're taking out all the variability of a highly outbred human population.
That's great, thanks. Another one for you, please, Daniel. How long do antibodies last in the body? Would multiple treatments of antibodies be needed for a COVID-19 treatment?
Potentially not. I mean, it could well be the case that you might only need a single injection, and that this would last anywhere between 10 to 20 weeks. Yeah, so it's a unique advantage of antibodies. Very long serum half-life. It can be further increased through engineering, and we believe that this single dose, long-lasting protection, which might last through a whole infection wave, is one of the unique advantages of this type of compound.
Great, that's fantastic. Ira, this one's for you. If I understood this right, the barcodes you developed for your pooled parallel testing is based on detecting the presence of a single gene from the 11 genes present in the COVID-19 genome. I'm curious, why out of all of the 11 genes you chose this single gene and not any of the others?
That's a good question. We chose the it's not even an entire gene. It's just a small chunk within a single gene, and this is the chunk that the current methodology from the CDC uses to detect. So we didn't want to choose a different section based on anything else. We just wanted to copy exactly what the most established existing methods are doing, but then just attach our little barcodes on and piggyback off their methodologies. We don't want to stray too far from what other people are doing, because it's just going to make it more difficult to more optimization required.
Great, thanks, Ira. Chris, I'm going to throw this one your way, please. Does the amount of exposure to COVID-19 affect the degree of illness, or is a single encounter likely to cause the same degree of illness?
Yeah, so it's a single encounter. It's just that chance, breathing in or touching the face, having touched a droplet somewhere, that's what gets you infected. The evidence is that people that have very mild disease, the virus replicates incredibly rapidly. So one virus particle very quickly becomes trillions. And the evidence is that there's comparable levels of virus being produced in people with very few symptoms as in people who end up in ICU.
Chris, is that why this particular virus is proving more difficult to control than the original SARS virus?
Yes, that's a really good point, Mara Jean. That's actually the thing that threw the whole plans that Australia and other countries had carefully developed to prepare for this kind of a pandemic. With SARS, the more severe your disease, the earlier you present to the clinic, and most people, almost everyone, did develop severe disease, or at least enough that they had quite a severe fever, and that happened before they started shedding virus. So that meant that they came to their doctor and they could be picked up and then diagnosed before they'd really shed the virus. The difference with COVID-2 is that you start shedding the virus 2 days on average before you develop symptoms. Even if you are going to go on and develop quite severe symptoms, you're already potentially shared it with a few of your close friends and relatives. Very clever of the virus, and that's part of why it spreads so terribly and why it's so difficult to pick up everyone that might be spreading it in the community.
And so with that in mind, you really do have to assume that anyone could have it, and accordingly the importance of the physical distancing measures.
Yep, that's why I'm on this side of the computer screen, folks. Not sharing information, but nothing else.
Thanks, Chris. This one's for you too, please. What specific evidence is there that SARS-CoV-2 came from bats?
Yes, that's very clear. So if you think about that sequence fingerprint process that Ira described so beautifully, that every time the virus copies it, it just every now and then there's a little change. So that provides a fingerprint, and you can see which virus is most closely related to the one that's circulating and jumped into that patient zero in China, in Wuhan. And at the moment, the closest relative, if you look at the entire virus genome, is a couple of viruses that were isolated from these Chinese horseshoe bats. Clearly, there are quite a few differences. So some time passed between that exact bat and the one that's spread there. But that's why we're pretty confident that it comes from bats. The other reason why we're confident that it comes from bats is that almost every great pandemic in the last 3,000 years has come from bats, the evidence is. And that's an interesting question.
Chris, is it true that bats don't get sick from the virus?
That appears to be the case. It's best shown for another cousin of this virus, MERS, that jumped from bats to camels to people, again in this millennium. People have deliberately infected bats with the MERS virus, and they get infected and they transmit the virus for weeks and weeks, but the bats don't develop the MERS, they don't become overtly ill. People have sequenced the genome of bats, and quite a few of the genes that our immune system and all of our cells, which have some intrinsic, what we call innate immune responses to viruses, that genetic circuitry has just been figured out in the last 5 to 10 years, a lot of that. And interestingly, in bats, quite a bit of that genetic circuitry has either been chucked out completely, it's missing from their genome, or it's acquired mutations that's tuned it way down. So it looks like bats essentially tolerate virus replication without making as vigorous an immune response as we do. And that may be why they're a reservoir for so many viruses, Hendra virus here in Australia, but it may also be part of the reason why these viruses, they get used to being in a host whose immune system doesn't respond too strongly. And then when they jump into us, our immune system is pretty powerful and it over responds and we get very sick.
That's really interesting and helpful. Thank you, Chris. Now, I am aware that we're fast approaching 11 a.m., but there are some really fantastic questions here, so I'd like to keep going if that's okay. But for those of you online who have other commitments, we will absolutely understand if you need to sign off at 11 o'clock on the dot, but otherwise, please do hang around. This one is for you, Daniel. Have you yet identified the receptors for the antibody variants being studied? In particular, are they mainly directed to the spike protein? And this one's from Anne Fletcher. Hi, Anne.
Yeah, so that's correct. So the antibodies are mainly directed against spike protein, although we're looking at also at some of the other coronavirus proteins. Because we're doing the process in vitro in the laboratory, we have complete control over this, right? So we know exactly what they bind, and you're absolutely right, it's predominantly spike protein.
Great, thank you. Another one for you, please, Daniel. How does our body produce antibodies? Is there a natural internal process, or must it always be introduced via vaccine?
Oh no, this is absolutely a natural process, right? And in fact, you know, without antibodies, you probably wouldn't be sitting here. I mean, we secrete, you know, many grams of antibodies, not just in serum, but also into, you know, the mucous membranes in, you know, in the gut, in the lung. I mean, these can be, many square meters, and you would be immediately getting infections without, you know, continuous protection through antibodies, which is an ongoing adaptive process throughout your life. And in fact, people who have and Chris can comment on that, people who have sort of genetic alterations that affect antibody development maturation often suffer from quite severe immunodeficiencies.
Chris, did you want to add anything there, perhaps both on the immunodeficiency and the autoimmune side?
You know, the enormous progress has been made in the last, 10 years in terms of understanding the genes that cause immune deficiency and the genes that contribute to autoimmune disease. And that's led to quite a few of these targeted therapies, most of them being antibodies like Humira that Daniel mentioned. The challenge for us now, and we're in a great position to do it, is to use all that information to really get to the heart of the matter of why the immune system's over-responding in the way it is in people. Can we target that over-response, but still leave the part that's controlling the virus?
And Chris, while we're there, another fantastic question here. How does COVID-19 affect people with autoimmune diseases, and how could the research initiatives help them?
The short answer is we don't have enough data to really make any conclusions about that. That kind of work is coming out in pieces, but one of the things that one always has to be very, very careful about is sort of starting to see a pattern with it not enough data, and then you get all excited. I'm as guilty as the next scientist about that, and then you get more data and the pattern's not true. So, having stubbed my toes on that trap a few times, that we'll wait until we've got a good solid data set before we're really sure.
Thanks, Chris. Ira, do we know if the different genetic mutations of the virus show different clinical outcomes in patients?
At this point, the large lineages circulating around the world are not really greatly functionally different. But there are some examples where people have done sort of theoretical studies of mutants that they've seen in individual patients. And those studies sort of predict that there's differences in infectious. These mutations are mostly in the spike protein that Daniel Chris has been talking about. But in terms of the large different branches of the tree that are out there in the population, there's no real difference between them at this point.
Okay, thank you for that. Chris, I'm going to throw this one to you. It reads, very interesting discussion. Thank you. Can you give perspective as to how your research parallels or differs from the work being trialled by Uni of Queensland and Mesoblast study, where survival rates for patients on respirators has been demonstrated to reach high levels?
Yeah, well, not quite sure which University of Queensland study that is, but the Mesoblast study, very preliminary. If it's one of those patterns which will either hold up or not hold up in due course, it would be wonderful if it was true. The experience that we've had both with this disease and with SARS and in so many diseases is that things that look promising in a very small study without a control group and without blinding the physicians to who goes on which arm of the study. Time and time again, medical science has learned that we get all excited about stuff because, someone got better and we think, a eureka moment. Then we do a proper controlled trial and they got better for some other reason. And so I just worry a little bit that can't jump the gun. Same issue is true for a whole series of things that we're keeping a close eye on. Many of you will have seen in the news about things that look very promising. Some of the anti-HIV drugs, for example, in anecdotal case studies look wonderful, but the first of the real proper randomized control trials reported out 2 weeks ago in New England Journal of Medicine, and it was impressively negative in terms of any benefit whatsoever, either in knocking down the virus or improving the health. So that's the tough side of science. You get your hopes up and then they get smashed by truth.
Thanks for that, Chris. This one I'll throw to you again, please, Chris. I've heard a lot about scientists to stop COVID-19, in USA. Are they also sharing data, or do you have any comment on this consortia?
You know, there's a lot of different consortia. I think one of the most wonderful things about this whole epidemic has been how the whole world, scientific community around the world has just put aside any petty jealousies, any national competition, even interlab. Everyone just is sharing their data. And in fact, I might throw to Daniel to just give an example of how quickly his team was able to get data shared from people around the world.
Yeah, so that's also my experience. I mean, you know, as early as mid-February, for instance, we were able to get, you know, structures and sequences of antibodies from groups out of China, for instance, who had done some of the very early research on the disease throughout January or so. And then that was very quickly followed up by researchers in the US and Europe. So, I mean, I agree. I mean, the amount of progress that has happened over the last 3 months is really quite astonishing. And much of it comes through open collaboration and also international collaboration. And it is, in a way, it's been scientifically, it's actually been a very exciting journey. And I think it's really changing the field in a very positive fashion as far as research is concerned.
And Ira, you might just tell people a little bit more about GISAID or GISAID. I don't know how you're supposed to pronounce it.
Yeah, so pretty early on, various different platforms for sharing genome sequences were established. GISAID, as Chris has mentioned, is one where a whole lot of people are sharing their coronavirus genome sequences. And last I checked, it was up to several thousand, something like 5,000. It's kind of the rate at which people are submitting data has kind of outgrown the database, and it kind of can't even keep up. So people are creating new databases and new, better ways to share the data. There's also a fantastic network been established called the Arctic Network, who are sharing protocols and tips and tricks for how to run genome sequencing of coronavirus in your own lab, and they're sharing them all over the world. And yeah, so it's really fantastic. It means that data is coming out of all the different countries in the world, and you know that tree that I showed is getting too large to handle.
Thanks for that, guys. Chris, this one I'll throw to you, please. I've read that the virus affects the carrying of oxygen in the blood, which is why body organs essentially suffer or shut down. Does this mean that people with, for example, anemia will have worse symptoms or outcomes as they already have low levels of oxygen being carried around the body, meaning they will, for example, have a worsening of symptoms such as shortness of breath?
Yes, I mean, you're right that, you know, one of the big problems is one of the first things you see on a CT scan of the lungs is in someone who's got moderate to severe COVID-19, or even some of the children who had asymptomatic COVID-19 in the very early part of the epidemic, is you see what they call a ground glass appearance change to the lungs. And what that is a diffuse pneumonia. A diffuse inflammatory response going out on the immune system reacting right through the lungs to the virus. And what that does is compromise that extraordinary capacity of oxygen to cross from the air we breathe into our blood. You know, the lungs are a miracle in terms of how efficient they are when they're healthy. So you're getting less oxygen into the blood. And then if you have fewer red cells to carry the oxygen, then that's a second strike. So that won't be, that'll be a big issue.
Thanks for that, Chris. Now we are approaching 11:10. Can I just check in with our panelists? What's our hard stop? Chris?
I've just texted the chairman of the board to let him know that this is even more important than he is, and that I'll call him as soon as we're done. Okay. I might not have a job in 15, but I think this is really important.
Okay, thank you, because we do have some brilliant questions coming through. Ira and Daniel, are you both okay?
Yeah,
About
yep.
10 minutes.
Great, thank you very much. Chris, for you, there's a lot of debate about children returning to school. Do you have an opinion on the safety of sending children back to school? Tricky one.
Well, I guess if it helps their parents stop killing them, that's a good thing. I think there's certainly a lot of parents that are at their wits' ends with homeschooling. Thankfully, my kids are past that stage. I was asked to battle with Wi-Fi and sons choking up when they go the Wi-Fi with YouTube. But no, but seriously, again, we don't yet have enough data on the extent to which the virus spreads in schools and children. But I think it is quite reasonable that as the government and all the experts that are advising government, I'm not an epidemiologist. There are some very good epidemiologists advising the government. I think the government has, you know, the proof is in the pudding. The government has managed this pandemic in Australia about as well as anywhere in the world. And I think that's a tribute both to our leaders, but also to the entire community of Australia, the fact that we all got behind the social distancing as well as we did and flattened that curve. I think for that same reason, at this stage, it's certainly clear with influenza, you really very quickly see patterns of outbreaks in schools. That's not obvious at all in this pandemic anywhere. And I think while we don't yet have good hard numbers on that, it does seem to be the case that it's a lower risk than other things. It's a balancing act because, and that's the great dilemma for the government and for all of us, is that on one hand, you could take it to extreme and say, look, we want to remove every risk of transmission, and that we can do that. But the social cost and the economic cost of going so draconian is, as we've all experienced in the last 6 weeks, you can do it for 4 weeks, you can do it for 6 weeks, but we can't do it forever. And we have to balance these risks. I think it is a reasonable balance to strike.
Okay, thanks for that. This is a spicy one. I'm going to give it to you, Chris. Any thoughts on recent comments by Nobel Prize winner Prof. Luc Montagnier, I've probably completely mispronounced that, asserting that COVID-19 could not have arisen naturally based on its genetic elements and its uncanny similarity to HIV?
Well, you know, I have huge respect for Professor Montagnier. You know, he did isolate the HIV/AIDS virus many years ago. I don't know what the data is he's referring to about similarity to HIV. Ira may want to speak to that, but certainly all the publications that I've looked at on the sequencing, it looks like a bat virus an awful lot. There is this curious piece of the virus that looks like the bat one, but looks almost identical to a virus that was just recently isolated out of pangolins, which is, you know, are one of the species that the world has been up in arms about, with China as a market for illegal trade in various pieces of pangolins. So the receptor binding domain part of the virus is 99% identical to pangolin virus, coronavirus. What I take from that is that somewhere there's been a recombination between two viruses, and but that's what coronaviruses always do. So everything that I see about the sequence of the virus looks like it's got all the fingerprints of natural selection at work, and I don't see any evidence for any kind of sinister germ warfare thing. I can't rule it out, but it just doesn't. There's no data for it whatsoever.
Yeah, maybe also
I got that wrong.
I think it's important to realize that there are probably over 10,000 different coronaviruses in bat, right? And you know, SARS-CoV-2 is about 96% identical to a bat coronavirus that was isolated as early as 2013. So there is no special explanation required while this thing jumped into the human population. And also just sort of a warning that this might well not be the last coronavirus virus that will jump into the human population, and that we need to be vigilant once this crisis subsides, right? Because the experience we had with SARS, that then very quickly public interest waned and we let our guard down. I mean, this must not happen again.
Thanks, Daniel. Did you want to add anything to that, Ira?
Well, I would say it's important to note that, you know, HIV is another RNA virus, and they share similar, you know, basic machinery, and this is why you would see similarity between coronavirus, HIV, or any other RNA virus out there. It doesn't necessarily mean that there's some sinister engineering behind that. It's just evolution.
Thanks for that. Okay, the next question, I'll throw this one to you, Chris, to begin with. Are those with primary immune deficiencies like CVID at higher risk than the rest of the population? And are we going to see a shortage on plasma for those who need it if the demand to treat COVID-19 increases?
That's a really good question. We don't have any hard data on whether people with primary immune deficiencies are going to get disease. There's just been a report come out online of 5 cases of selective antibody deficiency. They certainly, you can still develop the disease, but you know, how much more severe, or even if it's protective, there's still a debate about the extent to which some antibodies might be bad antibodies and contribute to the disease. So I think the prudent thing is to assume that if someone has a primary immune deficiency like CVID, that they will be at substantially increased risk. We certainly know that for many other viruses, and they, you know, all of us need to take, even that's part of why we all need to protect those people with all of our efforts in social distancing. In terms of the other part of that question, which is, could this cause a shortage of intravenous immunoglobulin, which people with primary immune deficiencies like CVID absolutely need? That's a real risk. I, in fact, understand that there's a trial that's starting and that's going to get quite a bit of coverage in the Wall Street Journal in the next couple of days in San Diego, in partnership with one of the big companies that sells intravenous gamma globulin. So this is the antibody fraction from healthy donors that's really absolutely life-saving for people with immune deficiency who can't make their own antibodies. Already in the last 5 years, and particularly in the last 2 years, the use of that antibody fraction for what we call off-label use for diseases that are not due to an inability to make antibodies and are not necessarily where there's any evidence that it's actually beneficial. But as that use has grown just through, you know, physicians feeling like they think it'll do some good, it's actually created already shortages for people that absolutely need it. And so my big concern is that, as we've seen already happen with chloroquine, that if the cart gets before the horse, that is, if physicians start to, you know, hear that, oh, this IVIG is great for COVID-19, when in reality there's no good clinical evidence for that, we could suddenly see a real shortage of IVIG for the people where it does really work and where they absolutely need it to survive. So I think all of us need to, you know, if we're in an opportunity to, you know, counter that tendency, we in one way or another, any of you that are physicians, please talk to your friends and say, wait until the randomized controlled trial's out, because there's already a shortage of IVIG, and we don't want to make matters worse for no benefit.
Thanks, Chris. Ira, this one's for you. Is Garvan researching the genetic makeup of the patients with severe COVID-19, so presumably those going into ICU?
So my lab, the work that I've referred to is specifically about sequencing the genome of the virus. But yeah, there is also obviously a lot of interest in the genetic makeup of patients and whether or not that may contribute to more severe or less severe disease responses in those patients. So there are laboratories at the Garvan who are very interested in this. I'm not directly involved in the work, but I think it's in the early stages, recruiting appropriate cohorts, etc. But it's obviously a very fascinating area of inquiry. So I would say keep an eye out for it, and we'll see how things go.
Great, thanks.
Chris might be able to give more information about.
Sorry, what was that?
The sequencing of the severe patient cohorts, Chris.
Yeah, that's work in progress. Again, you know, it's a matter of distilling the information from particularly cohorts who already have had their genome sequenced and are only in the last month or two have had an infection, and so that's work in progress.
Fantastic. Now, apologies, I've just realised there are a couple of questions that popped through on the chat function that I didn't clock. Here's one, Chris. Do you see a statistical correlation between numbers of testing and speed of opening up the economy?
I don't know if it's a statistical correlation, and I should preface it that I'm not an epidemiologist, but as Ira mentioned, there is you know, if we had a test that could be done for everybody on a regular basis to test for the virus, so that you could essentially have an antibody, have a test that showed that you're not shedding the virus, then that would obviously be one way to really open up the economy quickly. So there was actually quite a nice thought piece published a month ago from a very good cancer epidemiologist in the UK, saying that if we could just test everybody on a weekly basis, then we could just all get back to work. But that requires Ira to get his test to work. He's got it working for 200 at a time, but he would need to get it to work for 2 million at a time. So, Chris, so he's got work cut out for him.
Could I add, I think a great example comes from Korea, who had an early spike during the pandemic, but have got their situation well and truly under control. They've done it not necessarily by having severe lockdowns in there and you know, keeping people at home, keeping people out of work, but by very, very rigorous, frequent testing. So they've managed to, you know, keep their economy relatively open compared to countries in Europe and the US and Australia, and they've done it by massive testing efforts.
Fantastic, and that's a really good segue to the next question, which is how long, Ira and Chris, do you think it will be before we're able to test the Australian population and using this mass testing? No pressure, Ira.
Look, I mean, we're already doing sort of thousands of patients a day around Australia just with the existing tests. I would say the particular test that I'm developing, which, you know, is one of many competing methods to develop massive parallel testing, I would say is weeks or maybe months away from being, you know, able to be deployed in really serious diagnostic setting. But you know, these things move a little bit slower at the moment because getting reagents and things like this are a little bit slower than they normally would. But I would say in a few weeks or months, these things will be mature. And, you know, I would also expect that, you know, we might not be the first people to develop this. There's loads of competing initiatives to develop exactly this, and also just to scale up the existing test that we have. Yeah.
Great, thanks for that. Now, I've been advised that we do have a hard stop, unfortunately, at 11:30. So I'm just going to try to draw attention to perhaps just two final questions, if that's okay. There are some really lovely comments also coming through on the Q&A from one individual saying, just wanted to add a huge thank you for this great presentation, so informative and helpful. You guys are amazing, best of luck, and thanks to you all. There's lots of messages like that coming through for you, Chris, Daniel and Ira. So thank you everyone for your support. Before we conclude, I will be drawing your attention to an online survey. We'd really love to get your feedback, both positive and constructive, given that we may be having to host events like this for some time yet. The next question I'm going to throw to you, perhaps Chris and Ira. Is it possible people are not being detected via this standardized PCR test if they have acquired mutations in the primer binding regions? Have the regions been selected as they have a lower mutation rate than other parts of the genome? Sorry, I didn't do a very good job on that.
Yeah, so I was being a little bit overly simplistic to say that the current method only tests one region at a time. It actually tests 2 or 3 different regions, and so the chances of a mutation disrupting a primer site in all of those different regions at once is quite low. Likewise, because we're constantly collecting all this genomic data, getting the entire genome sequence from loads and loads of different patients all around the world, we'll quite quickly notice if there are mutations that are interfering with the regions that people are testing. So it's unlikely that will be a major problem, I would say.
Thanks, Ira. Daniel, are there any monoclonal antibodies already approved for therapy in order to fast track a treatment to be introduced?
Yeah, so, and Chris mentioned that as well. I mean, there are a couple of antibodies currently being investigated to sort of dampen an overactive immune system, and that includes both anti-IL-6, which is already FDA approved, and also some of the anti-TNFs, right? So it's a general, immunosuppressive agents that are already FDA approved. I mean, however, you need to keep in mind that those are really treating the symptoms of the disease and are not actually targeting the virus.
Thank you for that. Chris, this is a bit of a contentious one for you, please. Do you think that are researchers working on vaccines with no guarantee of success?
I'm very optimistic that one of the vaccine efforts will deliver the goods. There's never been more vaccine efforts dedicated to a single virus than there's over 100 efforts around the world. I got an update on a video conference with the National Academy of Science and Tony Fauci and the head of the Wellcome Trust last weekend. And, there's several of those are already in clinical trials. As Daniel said, we've never successfully made a vaccine against this class of viruses. They are clever viruses at evading the immune system. But those vaccine efforts use 50 different technologies that should bypass some of the virus's tricks. So I'm pretty optimistic just from mass action that some of those will be successful. And as Tony Fauci has said, we need a few of them to be successful because we're going to be very quickly in a situation of wanting to deploy those vaccines for everyone around the world. And so scarcity is going to be an issue. So having a few successful vaccines will be immensely helpful.
Thanks for that, Chris. So look, we are out of time. I'm just going to read out one of the final comments here. Chris, Daniel, Ira, who needs who when we have you? Thank you for all your work. Thank you for making today happen. May you all stay well, safe and alert. Really lovely message. I'm sorry we haven't been able to get to everyone's questions, but do hope you've enjoyed this session, and we'll certainly be doing more Zoom public seminars and events. So thank you so much for joining us. We will be putting up those survey questions, and we'd really love your responses to those questions. I think they're popping up hopefully on your screens right now. Won't take you very long. If you would like to donate to Garvan's research and indeed our COVID-19 research, we would love you to do so. We will be sharing with you a link to the Garvan donations page. Of course, I will emphasize that Garvan's pre-COVID-19 research is just as important as ever. Our research into cancer, osteoporosis, immune deficiencies, autoimmune diseases, diseases of aging. We need your support for all of our work more than ever. But of course, this is a really exciting opportunity for Garvan to contribute in a meaningful way to the fight against COVID-19. So if you're inspired by today, please do donate. But thank you. First and foremost, thank you to each and every one of you. You already support Garvan in so many ways. We're so grateful to you, our donors, our supporters, our partners for the future, our volunteers. You are what makes Garvan such a special place. So please keep physically distancing, stay safe, stay well. And if you do have any symptoms now, they have increased the testing capacity. So if you do have any symptoms, please do go to your nearest COVID-19 clinic, do ring ahead and make sure you get tested. But more than anything, stay well, and we hope to see you at the next event. Thank you so much. Thanks to our panel.
Thank you all.
Thank you.
Thanks.